I'm excited to tell you some about a project with Russ Popadetic on understanding genome evolution in endosymbionts and how horizontal transmission and recombination play into this process. There we go. So, as you are all well aware of, microbial symbionts have been pivotal in allowing eukaryotes to adapt to revolutionary new lifestyles and niches ever since the origination of eukaryotes with the evolution of chloroplast and mitochondrial organelles. So in order to perpetuate these associations over time, transmission mechanisms are needed to allow the symbionts and hosts to find each other each and every generation. And these need to be very reliable, otherwise you won't have these associations persist for very long. So there are two main modes of symbiont transmission. One is strict horizontal transmission, where symbionts are acquired through the environment, either from contemporary hosts or free-living bacterial populations. At the other end of the transmission mode possibilities, there is strict vertical transmission, where symbionts are inherited through the offspring, often through the germline or from brooded embryos. But there are also a variety of mixed modes that incorporate both of these strategies to various degrees. And as we learn more and more about various symbioses spanning the diversity of life, we found that these mixed modes are actually pretty common. I recently did a study, a meta-analysis of the whole body of literature on transmission modes, and found that these are actually pretty high frequency, about 30%. So, thinking about these transmission modes, they're actually not only important to enabling the perpetuation of these associations, they also are very important to determining the trajectory of genome evolution. So as Eric talked about previously, thank you for the introduction to this idea, endosymbiont genome evolution is largely determined by lifestyle. So here at this end of the spectrum, you have free-living bacteria that have large dynamic genomes, large population sizes, and then moving to this other end of the spectrum of host association, you have obligate organelles. And these are thought to have small population sizes restricted to host tissues, and gene flow from exogenous sources such as viruses, transposable elements, other bacteria is limited in these contexts. And so often how this is spoken about is a trajectory towards genome degradation as the association is perpetuated over time and restricted to the host tissues, so that ultimately you have this trajectory towards complete genome degradation, except for only the elements needed for the association. However, many associations don't exhibit this pattern, especially in the marine environment. And so we wanted to investigate what's going on in these other associations that don't have extreme genome reduction, but seem to have complete obligate host association. To do this, we used an ideal system that spans the transmission mode spectrum. These are chemosynthetic symbioses between gamma proteobacteria and bivalves. So at one end of the spectrum, we have mitillid hosts with species in the genus Bathymedalis. These symbionts, they all live inside of the gill cells, and they oxidize sulfide, fix carbon dioxide, and make ATP for the host. So they're absolutely required for host survival, and the association is probably good for the symbionts as well, because it enables access to reduced substrates in the presence of oxygen as well. We know that their transmission modes are distinct and conserved within these groups. So Bathymedalis species have strict horizontal transmission. They're acquired into the gills. Every generation, the larvae are hatched without symbionts. Species in the family Solomyidae, Solomia species in particular, have mixed mode transmission. So we know that they are contained within the ovary, transmitted through the oocytes, and can be found in larvae out in the environment. But genomic evidence indicates that there are frequent horizontal transmission events in these populations. And then at the far end of the spectrum, we have Symbiosize species, such as Calyptogena species, that are mostly vertical. So these are transmitted also through the germline, through spawned oocytes. And there are occasionally horizontal events in these groups, but they're very, very rare on evolutionary timescale, so I'm going to call them mostly vertical. And just to highlight the difference between these groups a little more, when you look at the 16S relationships of the symbionts, with free-living taxa also plotted on this tree, you'll see that up here we have the Sycamide symbiont species that are all each other's closest relatives, whereas the horizontally transmitted Baphomedalis symbionts and the Solomide symbionts are interspersed along the tree with free-living taxa mingling among them. So to investigate this question, we collected samples of these species from across the world from various habitats. The mytilids live in hydrothermal vents and in cold seeps. The Calyptogena species live in hydrothermal vents, and the Solomides are shallow water dwelling. Actually, you can find them off the coast here. It's an old collecting site of mine off of Narragansett. And we prepared nanopore and Illumina sequencing libraries, and I'd like to just briefly give a shout out to Evan Pepper for this. He generated the data and was pivotal in allowing this to go forward because we needed to make a lot of samples. So we also sequenced many individuals of each of these species to reconstruct the history of these populations. So after assembling the genomes, the first thing we did was we wanted to investigate genome size and just see if it's consistent with the other reported data and if it's consistent with what we know about transmission modes and the evolution of genome degradation. And indeed, the horizontally transmitted and mixed mode transmitted endosymbionts have large genomes, approximately the size of free-living sulfur oxidizing bacteria, which are around 3 to 4 megabases, whereas the Calyptogena species all have small, reduced genomes that are about half the size of free-living relatives. However, when we plot the phylogenies, or the genealogies of these populations for mitochondria and symbionts from host individuals and then draw other relationships between these topologies, we find that there isn't any evidence for strict co-speciation and co-cladogenesis because you see these lines crossing, and there have been many different transmission events that have broken up the linkage between mitochondria and symbionts that you would expect if they were always transmitted in the cytoplasm together. So this indicates that although we have genome reduction, this is happening in the face of a fair amount of horizontal transmission. However, I'll point out that this metric of looking at co-diversification is pretty sensitive to horizontal transmission. Even small amounts of horizontal transmission will erode any signal that you'd expect to see here. So looking at this a little closer, we wanted to look within these populations. So within the host tissues, there are millions, billions of symbiont cells in a population of its own. So looking at that a little more closely, comparing the within host diversity to between host diversity, we find that the horizontally transmitted and mixed mode transmitted symbionts are not differentiated. So host individuals, as you can see over here at their high horizontal transmission rates, they share genotypes between host individuals, which you would expect to see if symbionts are mixing between hosts. However, the Sycamide species are highly differentiated. Host individuals have completely different sets of genotypes. So this indicates that although horizontal transmission does occur, it's pretty rare in these groups. And looking at this a little more closely, at the allele frequency spectra within the host tissues, we see an even more striking pattern. So the mytilid species exhibit allele frequency spectra that look very close to the equilibrium expectations for selection and population size. They're like free-living populations. The mixed mode Solomia species have very crazy allele frequency spectra. They're super variable. Some of them look more like the free-living equilibrium expectation, but then other ones have these multiple modes, indicating that there were recent horizontal transmission events and recombination that happened that introduced new diversity into these populations. And then when we get down to the vertically transmitted vesicomyades, we see that they're nearly clonal. If you look at the y-axis here, there's only like 5 to 7 variants at each one of these frequency sites. So basically, these are clonal. There's not much going on in them. So thinking about this a little more in detail, if you have a horizontal transmission event, and to incorporate that diversity, recombination would help to enable it to enter that population and persist even through transmission bottlenecks, small population sizes. So we wanted to look at the recombination rates in these groups. So in doing that, this is rho times L, so the recombination rate times the recombination tract length. We see that the mitilids and the solomyids have high recombination rates, and the vesicomyades have significantly lower recombination rates. And so this will produce a pattern in these clonal genotypes of clonal interference, where you may have a beneficial allele, but then you may also have a deleterious allele, and there's no way of breaking these apart from one another. And so, under strict bottlenecks, you'll get the deleterious allele without being able to separate it, as opposed to recombinant haplotypes that can involve higher fitness. And when we look at these in a little more detail, this is the between host allele frequency spectrum. We see just this high frequency of low frequency alleles. This is also accentuated here on these trees, that you can see that the tips are very, very long relative to the internal nodes. So these are clonal haplotypes that are interfering with each other and preventing the evolution of fit genotypes. And then thinking about this on longer timescales about how this affects the whole pattern of genome evolution. These are whole genome alignments for the mitilids, the solomyids, and the vesicomyades. And you can see these two with mixed mode transmission and horizontal transmission, they're completely shuffled, recombined. They're very dynamic over time. There have been so many changes we can't even count. But then when you look at vesicomyades, these are largely syntenic, very few recombination events. And also, I'd like to mention that many recombination genes are missing from these genomes, so this explains some of the stasis. So basically, it looks like there is a threshold in horizontal transmission rate and recombination rate that below a certain point, you don't have enough gene flow and you don't have recombination to enable large dynamic genomes to persist, and you basically start the march towards genome reduction and a reduced static genome state. And the last thing I'd like to mention is that I think we should redefine this model to, instead of being a trajectory towards genome degradation, think of it more as there's certain stopping points along the way. Depending on your horizontal transmission rate and your recombination rate, any of these might be a stable equilibrium. And the vesicomyades have been around for tens of millions of years, the solomyids hundreds of millions of years, and their symbiotes have not degraded. So with that, I'd like to thank everyone who helped out with this project. Thank you for your attention and welcome me.